This is cellular respiration. There's a lot to go over, so let's hurry up. So there's two types. We have aerobic, which is oxygen, and anaerobic, and anaerobic, which is without oxygen. Fermentation happens without oxygen. Aerobic respiration happens with oxygen. So components of cellular respiration, photosynthesis. We went over photosynthesis, but cellular respiration is almost the opposite. So we're going to use sugar, what we made in what's the product of photosynthesis, and oxygen, which is another product, to make carbon dioxide, which is what's needed for photosynthesis, and water, which is what's also needed for photosynthesis to make ATP energy. So they are opposites of each other. So just remember that when thinking of their formulas. Energy flow and chemical cycle. So energy flow, we got photosynthesis will make glucose and oxygen, which is used in cellular respiration to make ATP and what we need, and we give water and CO2 back to the plant. It's a cycle. The plant gives us the stuff we need, we give the plant what it needs. So here we have we can split it up in three stages: glycolysis, Krebs, and electron transport chain. Glycolysis goes from glucose to pyruvate. Krebs will produce the things we need for the electron transport chain, and oxidative phosphorylation. We also have a mini step right here when we want to go from the cytoplasm to the mitochondria. So ATP, why is ATP important? It's adenosine triphosphate because it has 3 phosphates. And when you want to use ATP, you're going to break one of the phosphates to make adenosine diphosphate. ATP is made when energy is released. So we create it during cellular respiration. ATP carries, stores, and releases energy. This is why we need it, because it deals with energy. No phosphate disappearing act. So there is a law in thermodynamics. Thermodynamics that says nothing is created or destroyed, but rearranged. So we're not destroying phosphates. We're not making phosphates disappear. When we break it, we're going to use it again to make another ATP. Okay.
Yeah, and energy is within the bonds of...
Yeah, and those are...
That's why when we break it and become ADP, we use that breaking of the bond to release that energy to get used for something else.
Exactly. So glycogen is the splitting of sugar. So sugar, we want to split it into what? Doesn't this look like let me what's it? Triphosphoglycerate? Oh no, this is not CO2. No, yeah, this is just carbon.
Are you on slide 8?
Yes.
Okay, so first step is glycolysis, and we just basically take glucose, a 6-carbon molecule, and just split it in half.
And then we get the ATP and use the phosphorus from the ATP
So we do actually invest in some ATP to actually break the glucose.
To break it and then get the phosphorus we need to make CCCP. 3CP. Wait, is this what reduction is? We remove the O2? Do you think?
Let's see.
3G3P?
I just... Which process?
Remember photosynthesis?
For the Calvin cycle?
For the, Calvin cycle, when we reduced... Do you think that's what happened? Let's just not think of it too hard. This is something different.
I'll need to actually look much more into it. But
Yeah, you can tell us the answer later.
Focusing on glycolysis, the main purposes of this step is to just split the glucose in half.
Split it in half. Okay. So now we're gonna wait for the payout, energy harvesting. So we're gonna harvest some energy. So, and then the electrons we get will be transported with NADH. So we're gonna harvest. NAD plus will turn into NADH. Why? Because this H will have electrons, which will change the charge of this. This hydrogen will have electrons that will change the overall charge of this molecule. So we'll have electrons that we can use later on. NAD plus is a Uber. So we'll pick up electrons and drop them up and look for more electrons. So now we want to get your payoff. Now we're going to cut this. Now we're going to get rid of the phosphorus. And that will turn back ADP back to ATP. So we get some ATP out of glycolysis. And then we have 3 carbons at the end. So basically what we are doing this is to get here 3 carbons. 3 carbons. 2 ATP. What's made? 4 ATP molecules, but we do use 2 of them. We use 2 ATP to get 4. That's pretty good. 2 NADH molecules would transport electrons and 2 molecules of pyruvate. So this is every time we break 1 glucose. But every time this happens, we're probably breaking billions of glucose molecules like right now. Okay, now this is happening in the cytoplasm. Remember this. This is happening in the cytoplasm, not in the mitochondria. But to enter the mitochondria, we need to do this part, which is called CoA, I think. Something that's our end product. But so what we're gonna do? Link between glycolysis and citric acid cycle, or Krebs cycle. You give some. Okay, we're gonna remove the 1 carbon. So we're now with 2 carbons. Then we're going to get these 2 carbons, NAD+, and we're going to get more electrons out of that, right? More energy, more electron. We're going to harvest more energy, and we're going to get this coenzyme A, or CoA, and this carbon will attach, forming adenyl CoA. So what's made? 2 NADH and 2 CO2S. And this is we made adenyl, which is 2 carbons, CoA. Now, this is what is useful. Oh my god, so much stuff. This is what's useful. Do you want to talk for a while?
I can talk a little bit about the Krebs cycle.
Yeah, I'll pick it up in the electron transport chain.
Okay, so after glycolysis and our little transition from glycolysis to the Krebs cycle, we now have to do. Let's see. Okay, so now we then actually carry on the electron from the CLA to get NADH and FADH2, which is another transporter for the electrons. And from that process, we also then remove carbon using the oxygen that we breathe in. And from this Krebs cycle, we end up making 4 carbon dioxide, 6 NADH, NADH and 2 FADH2. And two more ATPs. And then from there, the NADH will get transported into the electron transport chain, which is the last step in the main area where ATP is going to be made. And that's where the H from the NADH will get dropped off. The electron from there will get dropped off.
Yeah. So basically we'll remove another carbon. Do we remove all the carbons or just one of them? And we're left with COA. So I see 4 CO2s. And last time when we removed one, we got 2 CO2s.
Okay, so we go back to...
So I think we're going to remove all the carbons. Because we get 4 CO2.
That's basically what's happening.
Okay, and we make some ATP. Let's go.
Okay, so I think what's really kind of happening is we're trying to remove all carbons and producing CO2, and we're just really after that one hydrogen electron that's getting carried by the NADH.
Yeah, we only care about we probably only care about the FA. I think we only care about the NADH and FADH2, because if we're gonna... Let's talk about the electron transport chain. So this... I'm on slide 19 now. This just shows that the mitochondria looks like this to increase surface area so more things can fit. But here's the good stuff. I think a lot of you guys have seen this diagram already, but this really sums up the electron transport chain. It's important to me, I believe, for both cellular respiration and photosynthesis. But let's start. Okay, we're going to get NADH, and that'll transport electrons. These purple things are electrons. So this is why we're producing all this NADH and FADH2, because they both transport electrons. And that's what we're harnessing the energy from. All these steps have been to get electrons and to power them up, so we can use that energy to put it in our bonds. So when we get rid of that, we get a hydrogen. The hydrogen escapes to the top. Right? Escapes to one side. Basically, it'll go on one side. Then we're gonna add more electrons. And once we're here, how do we get water? Oh, no.
We get water because...
that's why we need oxygen.
So when the NADH goes to...
I remember.
Gets transported in between the membranes from the proteins. Hydrogen electron bonds just drop out into the proteins, then moves its way downward into the ATP synthesis. There it will be able to combine ADP and another phosphate to create an ATP, and the rest of the hydrogen is then combined with the oxygen to create H2O. And that's why we need oxygen. It's to create that chemical bond to
get rid of the hydrogen.
Yeah.
We don't need hydrogen anymore. So we'll use the oxygen to get it out of our bodies. Okay, that's interesting. So we're gonna I think this step is, I don't know what this, I'm confused why this is step 4. Because I would assume the hydrogen would go on top. It'll be 5. Yeah, I know, that's weird.
Step 4.
I would assume that hydrogen would get up here. It would turn the ATP synthase to combine ADP plus another phosphate to make ATP.
It gets the oxygen already.
Maybe. And then when we like spin,
hydrogen goes into the ATP synthesis, it's there ready for it to,
bond and then get the hell out. Okay, yeah. Quick recap. NADH, FADH2 transfer electrons. The electrons are going to harness their energy. The hydrogens move up. Then they go down through the ATP synthase and they rotate it. They're like a crane. What would you call it? Like, you have to rotate it to move.
Yeah, so it's like one of those revolving doors there. You go in and you kind of push the door and it revolves around and then you get inside.
Ah, yes, yeah, exactly. But you're moving it that allows the ADP plus phosphP to make ATP. So we're going to use these hydrogens. But then when we have these hydrogens, we have nothing to do with these hydrogens now. And then we make H2O, which is water. What's made? 32 ATPs made. Okay, now we're going to discuss what happens if you have no oxygen. And after that, we're finished.
Yes, this is anaerobic.
This is anaerobic. So,
process.
Occurs under anaerobic conditions, without oxygen, anaerobic harvest of energy from food of some cells. So this is what happens without oxygen. We can get alcohol from it and lactic acid. We use it for food and all sorts of stuff. So here, glycolysis, we don't need oxygen. So we always go glucose glycolysis to make pyruvate. But because we don't have oxygen, we can't go to the Krebs cycle. So there's no point. So this will turn into lactic acid. And lactic acid, we can use that as energy. So some organisms produce alcohol instead of lactic acid. Food can be made by microbial fermentation. So some will make the 2-carbon compound instead of pyruvate. Like what happens in what's it? Wait, I can go back. It's on slide
wait a minute, sorry.
Slide 13. Slide 13. Lactic acid. Lactic acid. I think yeast will make lactic acid, and that will turn into ethanol or alcohol. And how do we get the stuff we need? Because we don't make our own stuff. We're not photosynthetic. We don't get our stuff from the sun. So how can we get? We can get it from carbohydrates, which have the glucose we need for glycolysis. An example of a carbohydrate is potatoes. Fats, they're glycerol for glycolysis, and fatty acids for lactate CoA. I don't think for this class. Yeah, for this class, I don't think we'll know what lactacil CoA is made of, but probably is made up of some sort of fatty acid. We can get that from butter. And proteins, we can use that in glycolysis, Actyl CoA, and Krebs cycle for those amino acids. And we got some shrimp and meat there.
I think the test will mainly focus on the...
Yeah, the final, I'm just worried about the small details she'll put on the final. I'm hoping that she'll put broad questions that highlight the entirety instead of small detail questions. Because there's a lot to go over. We're finished. Bye-bye.